idea regarding that how omics can help you in understanding the drug discovery. Till now, from morning, you have got an very good idea about different multi-omics approaches, multi-omics studies, and how those are conducted, and those are that are used for different understanding of the data. But now here, so one example or application is always in the diagnostic approaches, or the understand that how that can be used for your diagnostic angles. But another application is always the drug discovery and how the best the drugs can work on. So what we know from our studies of different uh, pharmacokinetic studies and all, that the drug has the two kind of consequences. If we look at that, one is your drug action, that one drug tries to get identify its particular receptor, gets attached to it, and then either get transferred to the molecule where it will act upon, where it will get the or it will get the action done on that. So which helps you to get two things. One is your initial consequence of the drug receptor combination, and another thing is that how the biochemical effects that occur due to that consequence of those, the sequence or the chain of events that happen. So there are different ways and the mechanisms of the drug reaction. So you can tell about physical chemical reactions, or yeah, just if
Ankit,
yeah,
sorry to interrupt. I think your voice is a bit slow. Uh,
let me just remove this.
yeah, I think
Just
if
speak.
yeah, it is better now. Uh, can you speak now?
Yeah, it is
Yeah,
better now, right?
it's better. It's better. Please go
Yeah.
ahead.
So I'll just say, so the either your so different, just some drugs, what happen if you just apply this, just like for the dermatological disorders, that it will just help you in the membrane and all, or the have a different other effects. But we are what mostly interested into understanding of the cellular mechanism of drug action, and how omics studies can help us in deciphering those drugs or identifying those drugs used for the in the which works in the cellular mechanism. So that what happens is that it helps us to identify the effects, that how after inhibiting a particular thing can help in the sequence of the biochemical events. So we all know from different hypotheses, different reaction mechanisms, that the drug and the receptor is like of a, we know that the lock and chain and lock and key which gets attached to it, and when it will get fit, so there is a cavity in the receptor, will get attached to the protein, and then it will go. So the receptor can have different locations. One is your nucleus, it can be within the cytosol, it can be within the cell membranes. And what it works on, like the, if we say in the methods of how it works, one is your agonist or antagonist, which helps. Agonist means that it the actual molecule, it replicates the actual molecule, and which actual molecule would have bind with the particular protein or a receptor, but here the drug is going and that event is not. Another is your antagonist. They are, it can be inverse, that whatever action it was there, it is reversing the reaction. So also there is then the nature of the interactions can be either your van der Waals force or hydrogen bond or ionic interactions, which we generally know. So now there are the four types of receptors. One is your ionotropic receptors, what you know as the voltage gated channels, like the Na plus K plus gated channels. So here you can, if there is an imbalance, you can try to get up to those receptors. Second is your type 2 is your metabotropic receptors. So metabotropic receptors is the kind of like G coupled protein receptors, and all those important linked with certain key mechanisms. Type 3 is your enzyme linked receptors, which like the kinases and those inhibitors. And 4 is your which regulates your gene expression. So these are the major 4 types of receptors that we generally know, and we try to inhibit those, or we try to uh, help in the means to reduce the effect of that particular disease. So these are the 4 major receptors that we work on till now. Now the question is that how to choose a specific receptor, how to choose a specific target molecule. So this is the question, because we know that what we try to achieve when we give a drug is that it will have the effect on that particular said protein, but it won't give an off effect or the drug effect. So the what whenever there is a invention of the drug or identification of the novel drug, always there are studies related to the toxicity, which plays a major role in understanding. So how to choose this? So here I wanted to bring you the concept of the Indian mythology. We all know that how that Sanjeevani was asked Hanuman to take it, Hanumanji to take it to for the specific disease. But see, the concept remained the same, that how to choose that particular ligand or the particular molecule for that particular receptor that will act or work the best. So as of now, today's day, we know, and we are discussing, we will be discussing different concepts or terminologies like precision medicine or personalized medicine, or all the personalized approach. So what is the future goal of the target or the diseases are the personalized approach, or that specific target, or that specific ligand that can work on that specific person for that specific amount of disease. So that is the concept which remains still valid, and what we are still looking for that answer to try to find out those particular or particular receptor or the protein target molecule that can help us to do. So here the comes the roles of the omics technologies. So as we could see that the with the past 2 decades from the advent of the human genome sequencing technologies, starting from the next generation sequencing, human genome map, then comes the your mass spectrometry and the best proteome map, and all the advent of the technologies like the sequencing technologies as well as the mass spectrometry proteomics technologies, then the emerging metabolomics technologies. So all these are helping us to understand the disease are the mechanistic cues for a particular disease in the different context at the different level. So how we can use those data to computational integrate those, as it was mentioned previously, that how those integration can help us to understand the network proximity, that to today's world we all know that the single molecule cannot work, and how these integrated networks can help us to identify the novel targets. So these novel targets can be first, the it is using to identify our novel targets. Second thing what it will know is that our drug has any off effect on other proteins or not, or on these particular networks there are any off effect or not. So both the things are very much important or crucial to understand, and those multi-omics integration is helping us to do that. And then we will get, of course, our achieve our target for the precision therapeutics, or the targeted for the new one. So now if I just divide it into all the 4 levels, genomics, transcriptomics, proteomics, and metabolomics, to in a simplified way, because there are other layers of omics as well, as you all would have by now understood and will appreciate that. But here, like some of those in that individual level as well, not only at the integrated level but at the individual level as well, there have been roles of those particular omics technologies in understanding or in unraveling new drugs, and those has motivated the field, and the motivated is motivating us to let us try out that the integration will work as one of the best way to for the any therapeutic condition. So for example, here you are getting that this was using the genetic drug target networks to develop new drug hypothesis for major dispersive disorder. So this gave the concept of your druggable genome, that is that the genes that are known to encode proteins that are, so how it is telling is that, so you, as I mentioned, that there are four kinds of receptors that we are generally taking. So which part of the genes or which part of the genomes are related to those particular proteins that are being already used in clinical trials, or otherwise how we can have those genes which are uh, coding for the proteins that are in similar, structurally similar to that regions which are already used or belong to that families. So that gave the part of the druggable genome, concept of the druggable genome. So how it has been done is that they had drug target in interaction, then the gene wise and the pathway analysis and the jiva studies of the genome. As you all have known, I guess by now, the genome-wide association studies. So, what are the phenotypes or the genotypes association with for a particular phenotype? That is very important to understand. So that's why only knowing the mutation or the genes or the SNPs won't work, but also how much it is associated for with a particular phenotypic condition we need to understand. And then what we can do is that we can try to find out the drug and the targets and what are the expression levels in the different tissues. So this gives us an idea that how the drug would work with a certain genetic condition to a particular people and with other. So this is what the in brief the genotype and the phenotype. So whatever changes we measure in the genotype is not always reflected, and there some SNPs can be disease causing to some, but cannot be SNP cannot be no association. Some SNPs can be associated with disease, some SNPs cannot be associated with the disease. So for example, you see this SNP1, so it is uh, for the individuals, these SNPs are causing the disease, but here you can see that there is no disease. So some SNPs will cause disease to individuals, some will not. So this is how things work. So these are some of the to databases and some of those resources that you can use for different applications in the drug discovery, like the GWS catalog, central NCBI pharma GKB. So you can try to mine the disease genes, narrow down disease risk prediction, disease mechanisms, or mine SNP drug response associations or genotype studies for the association of the different diseases. So these are the different applications that you can do with these databases and these resources. So now this I'll just give you just an example that how of the targetable genomic alterations. So like this PCSK9 has been used as a target for development of a new generation of your hypolipidemic drugs. Similarly, in one kind of brain tumor, that is meningioma. So this SMO mutation has been identified as one of the few targetable genetic mutations. So since it is mostly heavily NF2 mutated tumor, but still SMO mutation is that one of those targetable genetic alterations that have been identified meningioma. So as I mentioned that there can be some particular genes or particular at the genomic level itself, you can identify certain targets. So this is one. So there is a drug called Metabolomics. So that has been started to utilize in the against the SMO mutation and is in now clinical trials. So this is how at the genomic levels itself identified, you can identify the particular mutations and can take it forward for your druggable target. Now the as transcriptomics is what? The transcriptomics is just so genomics is mostly static and it gives us the information related to mutations and the SNPs. But when we start moving on with the transcriptomics, so here there are some changes or some changes in the means of the abundance level changes in the study in the particular cellular level, and which helps you further takes you to the expression level. So here what is that the role of the transcriptomics has been very much crucial for not only for identifying but for understand the potential adverse effects of the drugs in early stage. So whenever what we have done is that with the GWAS and the QTL studies and also these genetic and the genotype phenotype association studies, we could understood that some mutations and some associations of the drug target interactions. Now when we are giving the particular drug to that particular gene, so what is the expression level changes, so that helps, that transcriptomics helps us to understand that particular it in very early stage that there is any off target effect or not, whether that particular gene is being targeted or there is certain other adverse effects. So this is where role of transcriptomics comes very crucial. So the transcriptomics helps us to get the disease mechanism, mode of action of the compounds moving from disease genes, and these are some of those and this is very important to understand the early prediction of adversary drug effects. So these georepository and the array express and the expression atlas are all those databases from where you can use the transcriptomics data, and there are numerous studies you would find that treating your cells with a particular drug and how it is affecting changes, so you can analyze your data and can already retrieve those results before you move on for designing your studies. So this is the drug matrix, one of those important database which gives you the toxic effects for any particular drug and what is those adverse effects. So if you are designing or going ahead to target or brings or use some drugs for your particular disease or studies, if you want to research, so you can have a go back and look at it, that what are the reports. Also there are different, you might know that there are like drug bank or pharmaceutical which also gives you an overall idea about, and the chemical toxicogenomics database CTDs, which gives you idea about how the what is the toxicity effects of those particular drugs. So how so just to give a brief overview how the transcriptomics would play a role in understanding for the drug diseases. So far is a drug, you will get the total mRNA, and as you have know that it is both microRNA and RNA-seq, so you will obtain those. So here you will get the biological samples treated with potential activation of the drugs and try to analyze. So you will analyze both the disease condition, healthy condition, and the one with the treated condition. So what are the changes you can easily get after you analyze the data and can interpret accordingly. So here are the some of those examples how the transcriptomics studies have led to identified the lead optimization in the drug discovery studies. So this is one of the examples. So here, as I mentioned, that you can, after inducing the drugs, you can do the transcriptomics and then can try to find out those ratios. So these are the targets like ROS1, EGFR, PDA, 10A, MTP, etc., etc. So these have been these are in the therapeutic area, both oncology, neuroscience, and the results, the selectivity and non-target, and useful, and the decision was go or no go. So here you can see that PD 10A has been written to be no-go for certain compounds. So this helps you to understand that which compounds you can take forward for targeting certain genes or certain molecules for certain uh, therapeutic areas. So now the advancement is the single cell RNA sequencing, and from the RNA sequencing we are going to the single cell RNA sequencing. Now why and how the single cell RNA sequencing is important is that the genes or the tumors, if for mostly if in the case of tumors, if you try to look on, so that it is very much kind of diverse and the tumor heterogeneity is stays, and the cells or the cells makeup of that particular tissues is quite different. The most important thing what is different is the immune environment and the immune cells, what is makes the constituent of those. So, and the targets that can be taken forward for the immunotherapy based treatments that remains unknown. And whenever the whole RNA is RNA-seq is performed or we try to capture it, so it becomes lost. So what happens in the single cell RNA technologies is that when we do the single cell RNA sequencing, so we then try to map the cells with, for example, if we try to map that what kind of immune cells are there, or what kind of particular cells are there, then we get a very good map of that particular cell type. And if we see that there have been some kind of cells that are immune cells present against particular targets, or having an enriched population of that cells, then we can take those as a method for the target. So for example, this has paper has shown that the single cell analysis of human glioma and the immune cells identified S100A4 as an immunotherapy target. So they had nominated that this particular S100A4 as a target for the immunotherapy. And after you do that, then you can always try to go for the animal model studies and all those model studies. So this is another good, that the charting the tumor antigen maps. So single cell genomics is helping to understand those tumor antigen maps. So already in the immunosuppressive environment, if we get some hint and ideas that can be targeted, it is always useful. So now moving on from the transcriptomics is the at the proteomics level. So proteomics, we know that it's a protein. So after mostly what we talk about as the MS based proteomics, mass spectrometry based proteomics, that is different from your microarray based proteomics, as Dr. Arup said. So this is how happens is that the cells, the proteins. So what we generally do is looking at the bottom up proteomics approach, or we digest our proteins into peptides and then try to identify. But the major limitation here, what remains, is the coverage, that we cannot identify all the proteins in a mass spectrometry based thing, but we can do in a gene based studies. In the microarray, we can have all the 20,000 genes expression. But still, with the increase in the instrumentation, so now we can detect around 8,000 proteins. And also you can see that there are numerous studies you can find out, that have been using proteomic studies to find out the different kinds of biomarkers. So what we can use from the proteome profiling, so this is a concept of chemoproteomics, or the which helps us to understand in a drug target interactions and the off target engagement of those. So what is that? So here what we do is what is generally done is that the proteins are labeled with some fluorescent labeled molecule which binds to the protein and doesn't let the drug to bind. So then what happens is that you will get that the proteome with the drug and the no drug. So what is that effect of those having the drugs and not the drugs? You can easily get to it. So to identify your different, so what will happen is that if your target molecules or the receptors are blocked with the certain these certain molecules with fluorochrome, then what will happen? Your drug cannot go and bind to that receptor or the protein molecule. But if it gets bind, is it getting bind to any other molecule or not, any other proteins or not? Then what you will get that off target, what is the effect of that particular drug on your particular cellular level or the machinery, that how your what is their off target effect. So this is called your activity based enzyme proteomic profiling, also for different kinases, enzymes and all, by binding with ATPs and all, it performs, so to find out what are those effects. So there are also numerous technologies which are happening, that which doesn't change the protein's structure and nature, and can in an in vivo way can help you to understand this kind of things. So this is of the target drug selectivity, that how much in an entire proteome specific off targets can be linked to the toxicity outcome. So if your drug is also when it is your actual target molecule is blocked, but still your drug is going and attaching with any other protein and showing difference in the proteome, then it will definitely have some toxicity. And otherwise, we will have the changes in the functional proteome you can link to disease or performance of a drug, and the target proteins of drugs with unknown mode using unbiased protein profiling. So we can always try to find out those proteins. And then there's a concept of the drug repurposing. Also, the protein interactions helps us to understand the different roles of those particular proteins. So again, the PPINs or the protein interactions can be of your is2 hybrid system, as well as affinity purification mass spectrometry or co-fractionation. Using these are the two MS based approaches, and where it is the is hybrid system. So what happens is that we can get the networks of the proteins or the whichever target molecules that we are choosing, that how it is interacting. So this is one of those studies which showing that this is when the proteins are interacted, when human based proteome interactome maps or the all those protein interaction networks could be built up. So there are some of those nodes which you can see that this is your drug target nodes and these are the target nodes. So by choosing the specific drug target nodes and the target nodes as well, you can try to find out that targeting which particular proteins or which group of proteins can lead you to the desired effect, and which if you try to find out in the non drug target nodes, then it won't work. So which particular so this is an means this help us to narrow down our the receptors or the proteins that we are looking for targets, as well as the screening of the compounds. You will be choosing very less amount of compounds, less number of compounds to target for that particular nodes, and it will have an overall impact in understanding in this drug discovery process. So these are some of the resources for the proteomics, like the Pride and the Proteomics DB, from where you can download your proteomics data, and first as you will learn the analysis, and then can do and then can try to find out the protein interaction maps, like using string or and using protein chemical interaction links like stitch. So you can do that and try to find out that how protein and the chemical compounds are reacting, and then what can be their association and what can be their nodes, and also the human protein atlas and all for the how much is the protein level expression in your particular region. So this is another example is the drug repurposing. So this is an important concept, that how so what is, so we know that the timeline of the drug discovery takes around 25 it's around minimum around 15 to 20 hours sorry years to for going on to identification, lead optimization, then your uh, different phase 1, phase 2, phase 3 and all, then the approval, FDA approvals. But what we can do is that if any drug has been approved for certain other diseases, can we use it for those diseases? I think during the COVID time it was very much in uh, in this process, that how to repurpose those drugs. So this is an example of how those drugs can be used for different for the original, like it has been for the antihistamines or anti-neoplastic or anti-cancer
Hello,
drugs,
sir.
can be used for the different uh, diseases, like the communicable diseases like malaria, tuberculosis. So though it has been approved for certain these diseases, but it can be taken forward and can be used in different approaches. So these are some of the examples of the how systems biology or the omics based studies are helping to identify the drugs and helping to take it forward before going for clinical trials and all. So here you can see that digoxin, which is a tablet for the heart diseases or the drug generally used for
Uh,
heart diseases. Yeah.
Hello, Ankit.
Yeah.
Yeah, we have around 5 minutes left. If
Yeah.
we can
Yeah.
wrap up the talk.
I'll wrap up. I'll wrap it up. Yeah. Thank you. Yeah. So this is helping you to potential therapy in the medulloblastoma, which is a kind of a brain tumor. Again, the proteomic analysis helps you to understand the different pathways, the novel pathways that can be you tried out for the against the disease, and then for that particular pathways, any protein and the drugs that have been already discovered can be used. So this was all about proteomics, from where you can get knowledge about the chemoproteomics, or to find out the off target and the protein interaction maps, that how the protein nodes are, the drug nodes are interconnected, or how much those proteins can be and also by doing the differential expression, definitely we'll find out those proteins that are involved, and the pathways will give you that which particular uh, pathways are linked to that particular disease, and then you will try to identify the particular set of drugs. And lastly, drug repurposing based approaches would help you in understanding, and also after you apply that drug in your cell model or an animal model, what is that proteome level changes. But the transcriptomics level helps us to understand in a much better way, as the genes expression we can study of all the genes, but proteome coverage lacks a little bit in that manner. And last is the metabolomics, which is the emergent field. So if we look at the steadiness, or the means dynamicness, so the last so PTMs proteins have much things. So not only that protein, the PTMs also play a role in the study. It's a post and or mRNAs, we know that the different spliceosomes and all different plays a particular role, but the metabolomics are mostly remain to the at the phenotypic level of the genome, but the metabolomic and also the whatever final phenotype what we visible is in the form of the metabolites of the metabolomic. So this is how a natural human metabolomic map or the pathways look like. But what we can how metabolomics is also helping is to find out those, is that the metabolomic analysis using blood, urine. So whenever we give a particular drug, so drug also being a this in a metabolite, we can try to find out that how the drug have been into the different organs. So whenever so we know that it is a complex system. Whenever you take a drug, it will go through the pass through the digestive system, have some metabolism there. So what is that till the effector organ it reaches, what is the level of that, and what are the effects it is making at the each organ level. So this we can study using the blood, urine, and the so and the different biofluids. So what are the metabolomic changes, and what are the different and also the microbiome microbiota interactions between the human, what is happening, and then we can easily evaluate the safetiness of this. So here one of the studies which has used the tyrosine kinase inhibitor therapeutic response. So once the tyrosine kinase inhibitor has been given with your panel of the metabolic biomarkers in the CML patients, and these are some of the resources like the human metabolome database, mass bank metabolites, and all those where you are the repositories, and you can find out the different metabolomics based studies. So this is another thing which metabolites or the metabolomics or the phenotypes or the phenomics, which gives you to identify the lifestyle associated changes. So not only the genomic factor or the proteomic factor what leads the consequence of a drug to act on or not, also the lifestyle associated changes plays a huge important role. So these are highlighted these two recent papers which has tried to identify that how lifestyle associated changes can be used, as well as uh, healthy lifestyle and the metabolomics daily monitoring can help you understand that how a patient is working in a pre-diabetic stage or at the diabetic stage. So this is one of the important, this is one of the projects running in the Stanford University for the personalized omic profiling, or it is known as the integrated personalized omic profiling. So here it is the longitudinal studies that they are doing for going for a person who are in Delhi and sampling Delhi, both in a stressed state, not stressed, happy state, disease state. So everything is mapping. So how the changes are going on at the genome level, transcriptome level, proteome level, and the metabolome level, that will decide on the course of the for future treatment or this thing. So this is the concept of the precision medicine or the personalized medicine, which helps you to tailor the disease prevention and treatment, taking into account once both genetic changes, all those changes, as well as the how the uh, what are the lifestyle associated changes. So this is what we are aiming for, or what we are looking for to go forward, and which helps. So finally is that these three patients have the same disease, but on the basis of their profiling, genomic, transcriptomics, proteomics, or metabolomics, so we will diagnose the different biomarkers, the different receptors, if that person is having that particular receptor or not, and on the basis of those it will determine the drugs, and then you will, all the three will give a positive effect. But if this drug is administered to these two persons, then you will not get any effect, and it will become, for example, in cancer, the resistant and all, which was a very classical case of the breast cancers, with that drugs being used for a particular receptor did not work on other patients. So this is what we should look for, and we should try to, through these omics studies, we could achieve for. So to summarize, so this should have, a drug should have best on target response and no off target response, and helping identifying novel drug targets, screening drug efficacies, drug toxicities, and also it is facilitating the development of the precision medicine or the personalized medicine. So we can, the robust instrumentation and the data analysis, we can finally lead to the identification of what medication to be used for a particular person. But there are many challenges along with the regulatory approvals and all, and it is just an rather a concept or idea which needs to move very far to achieve this in reality. But omics based studies can definitely help in achieving that. So these are the references, and thank you to the organizing team for giving this opportunity, and for you for listening to it. So if you have any question, otherwise if it is late, I can take it. If you have any question in chat, and I can take it later as well. Yeah, thank you.